6 years now.
Okay, all right. So Paul Sigala joined us 6 years ago at the University of Utah. He got his undergraduate degree at the University of California, San Diego, where we probably all wish we were right now for a little bit of warmth. He then went on to do his doctoral training at Stanford in the lab of Dan Hirschlag, and then did a postdoctoral fellowship at WashU in the lab of Dan Goldberg, where at that time I think is where he became interested in malaria and started his work on heme metabolism in malaria. And today his talk, as you can see, is 50 Shades of Iron, Adaptive Heme Metabolism in Malaria Parasites. And I will monitor questions in the chat if you have questions. Paul has agreed to have interruption, and so I will invite you to ask your question if you have a question. And thanks all for attending, and we'll let Paul start.
Great. Thanks, Diane. It's a pleasure to talk to you all today and tell you a bit about our work. Decided to focus, even though we work broadly on a number of different aspects of heme and iron metabolism and blood stage plasmodium, decided to try to focus on our heme work and give you sort of a broad perspective of how we got started in this area, where we've been, and where we're going here. And as Diane said, please interrupt with questions in the chat. We'll make for a more lively discussion, even if it means I maybe don't get to everything, that's okay. So malaria is really an ancient scourge of humanity that humans have co-evolved with, and even to this day remains a devastating global infectious disease, with the majority of malaria burden and deaths concentrated in tropical Africa. And my exposure and interest in malaria started between college and graduate school when I spent 2 years teaching high school chemistry in Ghana, West Africa, as a US Peace Corps volunteer. Of course, this was a malaria endemic region and a major risk, especially for young children living in the community that were there. And this was in the early 2000s, which was really an exciting time in a way, at least for the global health community, because this sort of ushered in more than a decade of reductions in global malaria mortality and morbidity due to a variety of metrics, including successful implementation of public health measures, mosquito bed nets, and the introduction of artemisinin combination therapies. And so over the early 2000s till sort of the mid 2000 teens, global malaria deaths reduced about 40% or so. But over the last 6 to 7 years, it's become clear that these reductions have stalled, and especially with disruptions and complications due in part to COVID-19 arrival in Africa. For the first time in many years now, that global malaria deaths have started to increase, and there's concern about both spread of drug-resistant malaria into Africa and ongoing disruptions to reducing these deaths. So malaria is caused by roughly five plasmodium species, the most virulent of which is Plasmodium falciparum. So this is a mosquito-borne eukaryotic single-celled parasite, the sporozoite form of which gets injected under the skin of a naive individual. These traffic to the liver where they invade a hepatocyte, and over 10 to 14 days differentiate into tens of thousands of asexual merozoites, which then rupture into the bloodstream to invade red blood cells in a cyclical 48-hour fashion. And this blood stage of the infection is where all of clinical symptoms of malaria manifest. And it's also the stage of the parasite that we are best equipped to culture ex vivo in a laboratory setting. And so most of my lab's efforts is concentrated on studying the sort of fundamental cellular biochemistry of blood stage parasites. At low levels, these asexual parasites differentiate into sexual gametocytes, which are the mosquito infectious form, and are taken back up into the mosquito to propagate the infection. And if I can get to it at the end of the talk, I'll show you a little bit of data that we've started to extend some of our studies collaboratively into the mosquito vector. So in general, my lab's interested in understanding fundamental organelle biology. These eukaryotic parasites have a mitochondrion. Most people are familiar with mitochondria. They also have a second key organelle that's a non-photosynthetic chloroplast-like organelle called the apicoplast. Both of these organelles have key metabolic pathways and are validated drug targets, including atovaquone that targets cytochrome B in the mitochondria and doxycycline that's thought to block ribosomal translation in the apicoplast. We're broadly interested in understanding the critical functions of these organelles and defining biochemical similarities and differences from the human cells that they infect. There's quite a bit in terms of challenges and opportunities here. In many cases, as you'll see there, Plasmodium is a divergent eukaryote that branched off evolutionarily early on from well-studied organisms like yeast and mammals, which means that there can be proteins with unusual sequence features that can be very challenging to understand functionally, and there can be very limited biological precedents. But these also present an opportunity, if we can understand those, to garner rich insights into parasite evolution and the selective forces that have shaped that, as well as to identify new parasite-specific targets. And so one of the stories I'll tell you today definitely aligns with these challenges and we hope opportunities. So sort of broadly speaking, what I'm going to tell you about today is our work to understand how malaria parasites acquire and use heme as an essential metabolic cofactor. And this has spanned a variety of projects. I've sort of sprinkled on here the key players of our heme team of past and present Sigala lab members that have contributed to this work that I'll tell you about today, several of which are on the call right now. So it's been recognized for really over a century that heme and iron metabolism is really central to blood stage malaria. Of course, Plasmodium parasites infect red blood cells, which are the most heme and iron rich cell in the human body. And over 48 hours go from a small fraction of the size of that red cell all the way to nearly the entirety of that red cell volume. And they make space for themselves by ingesting most of the hemoglobin in the red cell into an acidic vacuole, and then using a cascade of proteases to break down that hemoglobin, both to utilize amino acids for their own needs, and it also liberates vast amounts of free heme. Now that heme is potentially cytotoxic, and so one major mechanism for detoxification, really the major mechanism for detoxification, is that heme is biomineralized in that acidic vacuole. So shown here on the lower left is a false cover electron micrograph, where in red is the host red blood cell, in gray is the outline of the mature parasite, and then in brown here is the digestive vacuole with those clear contrast-rich hemosome crystals that can be viewed within that vacuole. These really consist of a repeating subunit of heme-heme dimers where the propionates sort of reciprocally coordinate a neighboring heme monomer and then stack in that regard. Hemosome is, although best studied in the context of Plasmodium, is not unique to Plasmodium, and in fact other hematophagous organisms such as schistosomes and Rhodnius prolixus are also known to form hemosome, although in many cases the morphology appears to be more of aggregates than of distinct crystals, although that can vary with the particular samples that are there. More recently, many of you will be familiar with Iqbal Hamza's work showing that even in the case of a mammalian system, when mice who are deficient in the heme transporter HRG1, that in splenic macrophages, his lab observed that the heme derived from turnover of red cells can form hemosome within that compartment as well. So not unique to Plasmodium and really a broad feature of blood-feeding cells and organisms. One of the aspects that I'm not going to have much time to talk about today, but I do want to mention briefly that's particularly fascinated us and appears to be unique to Plasmodium, is that the heme crystals, shown here to be static in this EM micrograph, in the context of live parasites, really rapidly tumble and move in that vacuolar environment. So this is real; these are real-time speeds showing that hemosome tumbling. This is right at the diffraction limit of these several hundred nanometer that can be resolved with these several hundred nanometer crystals. And so we have a graduate student who's working to understand the mechanism and physiological significance of this tumbling, both through experimental methods and also developing computational models to build up a predictive understanding of the physical features of that vacuole environment that confer this property of tumbling hemozone crystals. And maybe in a future seminar, I can talk more in depth about this really fascinating aspect of plasmodium biology.
Well, I'm going to interrupt you because I
Yeah.
can't resist the question.
Sure.
Can you load this vesicle with a bead, and does it roll like that too?
Yeah, we're just starting to do those experiments of, sort of vesicular, encapsulation of these so far. One thing we do know is that if you isolate, let me just move forward so I can get that tumbling video again. If you do isolate free hemozone, it doesn't appear to tumble. And so the next question is, there are aspects of that are an emergent property of the boundary effects of a charged lipid bilayer that's there. Hemozone crystals, because they're sort of oriented ferric iron or paramagnetic. And so these are all aspects of the system computationally and experimentally that we're hoping to better understand how these sort of overall contribute to the observed motion.
Thanks.
Yeah. And so historically, heme metabolism has been a tremendously rich target for antimalarials. So compounds like chloroquine that are thought to block detoxification of free heme into crystalline hemozone have been one of the most successful antimalarials of our time. Atovaquone, which inhibits cytochrome B in the mitochondria and is currently used for prophylaxis. And the frontline antimalarial artemisinin, that's really a prodrug that has an endoperoxide bridge that is reductively cleaved through interaction with a pool of heme derived from hemoglobin digestion. So this has really been a rich area of vulnerability and activity for antimalarials. And so I guess we're motivated by the general belief that by understanding fundamental features of blood stage heme biochemistry, we can both optimize use of current antimalarials and potentially discover new powerful targets for novel therapies. So heme, of course, is not just this toxic byproduct of host hemoglobin digestion. These are eukaryotic cells. They have a mitochondrial electron transport chain and key cytochromes that require heme as an essential cofactor. And so early on, when we very first got into this, we were interested in understanding where do they derive heme? At least, when I was starting my lab or shortly before then, most of the focus had been that, well, host-derived heme is biomineralized in this vacuolar environment, but it was recognized at the same time that they expressed a complete heme biosynthesis pathway, and that at least early on was assumed to be essential. And so parasites were thought to just have evolved to sequester these pathways and still rely on heme synthesis. And so in this regard, they were sort of viewed, I'll use a metaphor here, of the proverbial sailor here that's surrounded by this, essential resource, and yet it's somehow inaccessible to them here, captured in this famous poem by Samuel Taylor Coleridge. And so early on, we set out to test this, understanding, well, all of these enzymes are there, and in Plasmodium, it has some similarities to mammalian cells, and that there's an ALA synthase in the mitochondrion, and then 4 enzymes that in mammalian cells, of course, are cytoplasmic, but in the parasite are targeted to this apicoplast organelle, have some features of fungi in that the CPOX is cytoplasmic, with the last 2 enzymes being back into the mitochondria. And as I said early on, roughly a decade ago, this had been assumed to be an essential pathway for blood stage parasites. We first wanted to probe activity by developing some metabolic labeling. And so we had developed a C13 labeling assay with labeled aminolevulinic acid, that's the first committed precursor, and showed that we could feed that to parasites and readily detect formation of 8 Dalton mass shifted C13 labeled heme, as well as upstream porphyrin intermediates along the way. So this is just comparing here extracts of uninfected red cells, and then when there's a parasite inside, that we could readily detect C13 labeling of heme, protoporphyrin 9, and coproporphyrin as well. And so the next test, of course, was to say, well, is this really essential? And so in collaboration with a second lab, Vigislav at Drexel, we were able to knock out the terminal parasite enzyme in this pathway, ferrochelatase, show that that specifically ablated C13 isotope incorporation into heme, showing that we had knocked out this pathway. Those parasites were viable, and then in head-to-head growth assays, they grew indistinguishably from the wild-type parasites, really emphasizing that at least in the blood stages, Plasmodium parasites don't require heme synthesis for growth. We had extended these studies using the inhibitor succinylacetone and at concentrations that clearly block isotopic labeling of heme and porphyrins. There was no impact on parasite growth. In work I don't have time to go into here that extended these studies, it's clear that they do rely on this pathway in the mosquito and liver stage growth, and that really can explain evolutionarily why this pathway has been retained. And so just some aspects here, what this suggests then is that they actually can scavenge a portion of host heme to meet metabolic needs, although we don't understand the mechanisms of that trafficking at all. There are no identifiable homologs of known heme transporters from other cells, including that HRG1 that Iqbal's lab has worked on extensively. We'd like to better understand how labile heme is distributed within the cell. These end up being fairly challenging questions to go after, but we've started deploying some of them Meet Ready's biosensors and are getting some initial hints of what these distributions may be. And that's an area that I hope to follow up more in maybe a future talk. And so for the rest of the talk today, what I'd like to do is tell you about our efforts to understand parasite heme utilization. We got interested in this because for an organism that's evolved to grow in literally the most heme-rich cell in the human body, there are relatively few identifiable heme proteins annotated in the parasite's genome. And so one can readily see clear homologs of mitochondrial cytochromes, including cytochrome B and C1 that are part of complex 3, cytochrome C, and then the COX-1 subunit of complex 4 of the respiratory chain. Beyond that, there's a divergent hemoxygenase we've worked on that binds but has lost the ability to degrade heme, and then several cytochrome B5 homologs that are essentially unstudied. And so we set out to really ask, what are the sort of core and essential uses of heme that the parasite relies on? And so just focusing on this electron transport chain, we know that parasites require heme to support the electron transport chain function. Early on, the best evidence of this came from the drug atovaquone, that's a selective cytochrome B inhibitor. And it turns out that parasites require that, as I'll show you, not so much for supporting ATP synthesis via mitochondrial oxidative phosphorylation. They have an ATP synthase. These subunits are dispensable in blood stage parasites. They can be knocked out. They grow just fine. They do require that for growth in the mosquito and liver, and I'll get back to that towards the end of my talk. So rather, and I should say for blood stages, they primarily source ATP through cytoplasmic glycolysis. And so for blood stage parasites, the major function of that electron transport chain is to oxidatively recycle the ubiquinone cofactor. So in fact, you can rescue parasite viability from atovaquone by adding in the soluble ubiquinone analog decylubiquinone, and these grow indefinitely. And they need this to support the function of various mitochondrial dehydrogenases, of which the most important is the dihydroorotate dehydrogenase that they need for pyrimidine synthesis to support DNA replication during blood stage growth. And so we set out to really think about directly probing mitochondrial cytochromes. Of course, 2 of these are encoded on the mitochondrial genome itself. So Plasmodium has one of the most reduced mitochondrial genomes for organisms that retain a mitochondrial genome. It's only 6 kilobases and it only encodes 3 proteins: cytochrome B, the COX-1 subunit, and also COX-3 as well. And so these aren't readily accessible to genome editing and molecular genetics. So rather, we focused on the known genes encoding cytochrome C1 and cytochrome C. And sort of the plot thickened a little for us when we realized that parasites had actually duplicated their cytochrome C gene and encoded 2 distinct paralogs, so 2 different genes that each appear to encode a cytochrome C. And we thought this was curious. It's not without precedent. So yeast and other fungi have gene duplication, although in yeast there appears to be a high degree of functional redundancy, and both copies have to be disrupted for respiratory deficiency. These are C-type cytochromes, which in contrast to most other heme proteins that non-covalently bind heme, cytochrome C and C1 use a conserved CXXCH motif where the two cysteines form thioether bonds to the heme, and that histidine provides one of the axial ligands. This covalent heme binding is not autocatalytic, but requires the activity of another mitochondrial enzyme, holocytochrome C synthase, that helps to bind that cytochrome C and position the bound heme to enable those thioether bond formations. And so, as I said, parasites encode two paralogs, and unlike yeast, they're quite different from each other. In fact, one of these is literally the most divergent eukaryotic cytochrome C that we're aware of. So I've made a sequence alignment here. In blue is just human cytochrome C. The more conserved plasmodium, I'm calling just cytochrome C, and that's 60% identical to human C. And that second copy I've termed cytochrome C2. The nomenclature for these cytochromes is a bit dizzying, so I hope that you're able to follow me here. And that cytochrome C2 only shares 30% sequence identity with the parasite in human cytochrome C. Now, all three of these have that conserved CXXCH motif expected for covalent heme attachment, where that histidine provides one of the axial ligands. All three also contain that conserved methionine residue that provides the second axial heme ligand. And I'm showing that here in just the structure for human cytochrome C above. Now, hemylation of cytochrome C, as I said, requires interaction with that HCCS enzyme, and that's from work from Bob Kranz's lab at WashU, has shown that, and other labs, I should say, that requires sequence just N-terminal to that CXXCH motif to be bound and recognized by that HCCS element for hemylation, and those key residues are conserved in both plasmodium homologs. But what really differs in this C2 is that sequence just N-terminal to the methionine has completely diverged. And that really caught our attention because that is extremely unusual and seemed incompatible with a conserved role for this cytochrome in the electron transport chain. And in fact, work done on yeast cytochrome C from several labs, including Bruce Bowler's lab in Montana, has shown that mutations in this region, especially loss of that lysine residue, are sufficient to destabilize axial coordination by methionine, and such that the methionine has a propensity to swing out from there. And this dramatically changes the reduction potential of that heme center and enhances the ability to bind peroxides and thus have increased peroxidase activity. So what we set out to do is basically ask, are both of these parasite cytochrome C homologs hemylated? What are their physical properties, especially of that unusual cytochrome C2? Are they both in fact mitochondrial? And which of them or both are essential for parasite growth and viability? So the first question we approached by reconstituting hemylation in using E. coli as sort of a model test tube here. So E. coli, of course, synthesizes heme, but it lacks cytoplasmic C-type cytochromes and uses a completely different maturation machinery that's in the periplasm, not in the cytoplasm. And so extensive work from a number of labs have shown that heterologous expression of a eukaryotic cytochrome C in E. coli, together with a holocytochrome C synthase, in general is sufficient to reconstitute hemylation of that cytochrome and can be read out with a simple chemiluminescent assay, where if these are expressed in E. coli, the apocytochrome C together with the HCCS, that in general is typically sufficient to form the holo-hemylated cytochrome C, which can be fractionated by SDS page, transferred to membrane, and an luminol-based assay exploits the intrinsic peroxidase activity of that exposed heme and leaves this overlapping signal of that heme because it stays covalently bound and thus co-migrates with the polypeptide in the SDS page gel. And so just showing you what that data looks like here by Western blot, if we just express human cytochrome C and E. coli, there's no detectable heme stain at all. But if we co-express it with the human HCCS, now we get robust heme stain signal indicating that there's heme attached to that cytochrome C. So we basically use this assay to co-express both of the parasite cytochrome C and E. coli together with the parasite HCCS and ask if one or both of them was in fact capable of hemolation. And that's what we saw. So just walking you through this data here, on the left is the more canonical cytochrome C, on the right, the more divergent C2. If we leave out the parasite HCCS, there's no detectable hemolation, but when we co-express it with the parasite HCCS, we get a robust heme signal in both cases. So despite those unusual sequence features, especially in the C2, at least from these reconstitution studies, it was clear that the parasite HCCS was capable of hemolating both proteins. This turns out to be a rather challenging question to pursue in parasites. These are not expressed at high levels in the blood stages, but we do have some evidence that supports that both are hemolated and expressed in blood stage parasites. So since we were able to recombinantly express these proteins in E. coli, we next moved to purify them and interrogate and compare and contrast what their physical properties were. Again, pointing out that these sequence features in that more divergent C2 suggested that these must have, this protein must have an altered properties relative to the conserved cytochrome C. So we expressed and purified both parasite cytochromes together with the human cytochrome C. All three eluted from a gel filtration column at the expected retention time for a folded globular monomer, with small differences that were consistent with the hierarchy of their molecular masses and thus sizes. In fact, when you overexpress these in E. coli, it turns them bright red from the bound heme that's there, and we're able to purify these to single band by SDS page. So the first test that we turned to were UV-vis absorbance spectra that can really give incisive information about the structural and coordination environment of that bound heme. And so here, just comparing human and the more conserved Plasmodium cytochrome C. So these are spectra of oxidized cytochrome C. We see a strong Sorey peak around 408 nanometers, indicative of that heme center there, and a smaller transition around 530 nanometers expected for an oxidized biaxial, so two axial heme ligands at that center. We can stably reduce both of those with sodium dithionite and the Sorey peak redshifts, and we get then the Q bands that show up strongly at 520 and 550 nanometers, again, as expected for reduced biaxial heme, and suggesting then that more conserved Plasmodium cytochrome C really has physical properties and structures very similar to the human protein. That more divergent C2, however, showed something a bit different. So rather than showing that 530 peak, rather there was just a small weak transition a little above 600 nanometers, which based on mutagenesis studies and a variety of heme proteins has suggested that there's only a single axial heme ligand stably formed in the oxidized state and the presence of an open pocket, right? At least that's a suggestion based on interpretation in these prior studies there. And so we thought, well, if it's true that it has an open pocket, can we add exogenous imidazole, and will that now form a stable axial ligand? And in fact, that's what we saw. So in the presence of excess imidazole, now we can shift that 602 peak to a 530 peak as expected for biaxial coordination that's there. We can stably reduce that divergent C2 with sodium dithionite to give similar 520, 550 peaks. However, unlike the more conserved Plasmodium cytochrome C and human C, that rapidly oxidizes within a few minutes back to that 602 peak. And so from that, that's sort of consistent with the general picture that there's heightened accessibility to molecular oxygen that can get in there and oxidize that heme center. And so we've extended these to some EPR, electron paramagnetic resonance spectroscopy studies to better understand the structural environment of that heme. So shown here is EPR spectrum of human cytochrome C. This is data from one of Bob Kranz's papers from a few years ago, showing the presence of then low spin ferric iron center and human cytochrome C here. And when we acquire spectrum of that more conserved cytochrome C, see spectral features that are very, very similar, again, consistent with a very similar structure and physical environment. That divergent C2, Dennis, I see your question. I'll get to it in just a second. That C2, however, has a much more complex EPR spectrum with high spin features here that are more consistent with a pentacoordinate, so only having a single axial coordination event there, although there are some weaker features here in the low spin region suggesting that maybe there's a distribution of structural states that's present there. Let's see, Dennis is asking, is a C2 stable to oxygen in the presence of imidazole? That's a good question. We've done those experiments. I think it's more stable, but we do eventually see that it does oxidize. So it's not completely protective, but does seem to stay reduced perhaps a bit longer. So just to summarize all of this, parasites have two divergent cytochrome C. The more conserved one has very similar properties to the human protein, but that more divergent one, which is extremely unusually divergent for a protein that is one of the most highly conserved proteins in eukaryotic mitochondria, that it appears to lack stable biaxial coordination, has an open binding pocket that's capable of binding exogenous ligand, and seems to have greater accessibility to that heme center. So we were curious to know what that structural environment might be. And so working with Chris Hill's lab, a structural biologist in the department, we were able to crystallize that C2 and determine an X-ray structure of it. Although all of our attempts have only resulted in a domain swap dimeric structure. So again, I showed you evidence that in solution, even this C2 behaves as a globular monomer. Yet when we crystallize it, and these crystals always form slowly over several weeks or months, they always crystallized as a dimer, that where the C-terminal helix of one cytochrome C had wrapped around to partition and basically assume the position of that corresponding helix in the monomeric form here. This is not a unique structure. This has been reported for canonical mammalian cytochrome C before, but is typically associated with specific conditions that involve the presence of detergents or certain alcohols, and has been of, I think, uncertain view as to what biological relevance it may have. In this case, despite extensive efforts, we've never gotten the monomer to crystallize. We suspect that that's telling us about conformational heterogeneity in that C-terminal helix. And though we don't think this is the biologically relevant form, this structure nonetheless has provided some useful insights, and I'll walk you through those briefly. So first of all, this divergent cytochrome C2, despite some insertions in multiple places and sequence divergence, nonetheless retains that conserved cytochrome C fold. So this is just a superposition from that dimeric structure with the human monomer that really says, if we set aside that this helix is really coming from the neighboring monomer, the overall positioning of that is as expected for a eukaryotic cytochrome C. Zooming in on the bound heme, it's covalently bound heme through these thioether bonds, and that conserved histidine is providing one of the axial ligands. Of course, formation of that domain swap dimer does perturb that axial ligand methionine. Again, that's observed even in canonical domain swap dimers, and so we don't think that that's biologically relevant, but at least is consistent with the overall properties. So Roland's asking if we tried crystallizing the C-term deletion. We've tried many different mutants and variants here. We've never gotten, for the C2, we've never gotten a monomeric form to crystallize. And so the last bit that's interesting, zooming in, and so Henry Winkers, who's Chris's graduate student who worked on this, so this is just a superposition between one of the two dimeric monomers from C2, shown in cyan, with the human one. And so what one can see here around this alpha helix is typically there is a kink after one helical turn where then that wraps around to where that axial methionine is, and then that C-terminal helix then partitions down into this pocket. In C2, the structure shows that there's an extra helical loop here that may move that pivot point out a bit further, and we hypothesize that that may contribute to destabilizing that axial coordination event, and thus predispose that portion of the structure to some conformational heterogeneity that may explain both the absence of a stable axial ligand, increased accessibility to molecular oxygen, and may disfavor then stable crystallization of a packed monomeric form. All right, so in the last little bit here, of course, we wanted to turn back to studies in parasites and say, okay, this is cool biochemistry, but what functionally is going on and which of these proteins do parasites rely on? So we used CRISPR-Cas9 to tag the endogenous gene for all of the C-type cytochromes in parasites to encode both a C-terminal hemagglutinin and epitope tag for localization and expression studies, and also a conditional knockdown system that's somewhat unique to Plasmodium, in which the 3' UTR is tagged with a repeat aptamer region that will tightly bind to the TET repressor that's expressed separately off the same plasmid that is integrated into the genome. TET repressor, of course, is not normally expressed in Plasmodium. That's fused to an effector DOSI such that in the presence of the non-toxic tetracycline analog ATC or anhydrotetracycline, the TET repressor is dissociated and there's normal translation. However, when ATC is washed out, that binds to this aptamer region and then sequesters those transcripts and turns translation off. So the bottom line is this is a TET on system. So plus ATC is normal translation, minus ATC translation off. So we use this system to localize all of these proteins. So both cytochrome C paralogs together with the single copy cytochrome C1 and that HCCS all localized to the mitochondrion. Of course, these are expected to be in the intermembrane space, but these epifluorescence images lack the resolution to confirm that. And so we're turning to some other studies to dissect that localization, but minimally confirms that these are all mitochondrial. We've done some IP immunoprecipitation mass spec studies that confirm expected interactions for mitochondrial proteins. So we first turned then to looking at which of these are essential for parasite growth. And so let me just walk you through what this looks like. So when we assay this, what we do is we synchronize parasites to the young early ring stage. And remember that they have a 48-hour life cycle in red cells where they divide and then rupture and then go out and reinvade. Mature red blood cells lack a nucleus. And so what we do is a simple flow cytometry assay when there's a parasite inside. Of course, now there is a nucleus and we can detect that DNA stain. And so we just plot the percentage of red cells in the culture that have a parasite inside. That's parasitemia. And for a synchronous culture with a 48-hour life cycle, we expect a stepwise increase in parasitemia every 2 days or 48 hours with each rupture and reinvasion event. And so that's just parasitemia on the y-axis and the number of days of growth on the x-axis. So plus ATC, where there's normal cytochrome C1 expression, those parasites expand and grow and parasitemia increases just fine. However, when we wash out ATC and can get reasonably good knockdown of cytochrome C1 expression, there's a slight delay, but basically after the second growth cycle, they fail to expand into a third. And by blood smear, we know that these are largely dead parasites. We observed a similar phenotype with that more conserved cytochrome C. So this is the one that has canonical features of a cytochrome C. Again, when we knock down its expression, parasites are not viable beyond the third day and die quickly. We were able to rescue parasites from those knockdowns by adding in that soluble ubiquinone analog, decyl ubiquinone, really confirming the sort of key role of the electron transport chain and functions of these proteins and oxidative recycling of ubiquinone there. However, we saw something very different for that divergent C2. So by knockdown, we washed out ATC, despite reasonable, though not completely stringent knockdown of detectable C2 expression, there was no impact on growth of those blood stage parasites. To rule out that there simply was just a small amount of protein still fulfilling an essential function, we made a clean deletion of that gene, and those also came back, were viable, have reproducibly grown with a very minor defect. We haven't complemented these yet. It's subtle, but there we think it's real. And we've started to do some growth competition assays to test that more fully that suggest that over extended growth times, that there is a fitness defect. These experiments are preliminary, but if true, would provide a basis for why parasites have held on to this gene, at least in the blood stages, that could explain a minor growth effect. You know, in biology, we like big phenotypes. We like to see we knock something down, things die. That's very gratifying. Of course, evolution operates on a much more stringent and subtle scale in many cases, and this could be one of those. However, in thinking about why parasites might retain this unusual cytochrome C2, we noted from transcriptional data that the mRNA for this gene was massively upregulated during mosquito stage growth. So as I said, these are just blood stage forms here, the ring to schizont. The gametocytes are the sexual form that's still in the blood stages, but the ookinete is one of the mosquito forms that's basically forms in the midgut of the mosquito and then burrows through the wall of that midgut to set up an oocyst. And so there was a greater than 10-fold increase in transcript levels, and we thought that that might suggest an enhanced role in the mosquito form. This was intriguing to us because it's known that fundamental mitochondrial biology undergoes a tremendous transformation in going from the asexual blood stages to the sexual and especially the mosquito vector forms. As I said, in the blood stages, parasites only require a low-level electron transport chain function to support oxidative ubiquinone recycling and not for supporting oxidative phosphorylation. However, that changes. And in the mosquito now, they're absolutely reliant on ATP synthase. Aspects of the TCA cycle and heme synthesis are all essential in that mosquito stage. And we thought this might be hinting at some special role for that cytochrome in the mosquitoes. As I said before, this destabilization of that axial ligand, at least in other eukaryotic cytochrome C, is known to open up that binding pocket and increase peroxidase activity. And in fact, that's a known transient property of mammalian cytochrome C during cellular apoptosis. And so we haven't yet tested, but our hypothesis is that this divergent C2 may function as a constitutive peroxidase. And we thought this could perhaps explain a key role in mosquitoes where there's enhanced electron transport chain flux that may increase oxidative stress. And this could be a key enzyme in oxidative protection in that role. And so we turned to studies with a collaborator, Fatini Sinis, who's at Johns Hopkins, to test a requirement for this unusual cytochrome for mosquito stage growth. So as I said, these experiments involved allowing mosquitoes to feed on blood stage parasites in which at low levels they've differentiated into these sexual gametocytes. Within the midgut of the mosquito, those mate to form the ookinete, burrow out of the midgut to form oocysts. And so this is just what that midgut looks like, and these sort of yellow circles are oocysts on the outside of that mosquito midgut. And then those form sporozoites, which then go to the salivary gland. And so our collaborator Fatini, basically, her lab counts oocysts on midguts of mosquitoes and then dissects out the salivary gland to count the number of sporozoites that have formed there. And so our hypothesis is that we might see a substantial defect. And in fact, this was one of those circumstances where I was absolutely sure that we were going to see a key defect in mosquito stage growth, but biology does its own thing. And those knockout parasites, when they fed the mosquito, formed oocysts and sporozoites just fine. And intriguingly, although it's somewhat subtle, we actually consistently saw subtle increases in the number of oocysts and the number of sporozoites that formed in those C2 knockout parasites relative to the parental line. Fatini thought that maybe the more permissive sort of lab cultured Anopheles stephensi mosquitoes that don't mount as robust of an immune response might be sort of skewing things. So she repeated these experiments with a more sort of wild or field applicable Anopheles gambiae mosquitoes and essentially saw the same result. Again, slightly higher oocyst counts in the knockouts and slightly higher sporozoite counts as well, suggesting that this effect is real. We haven't complemented this yet. Of course, we would need to do that to rigorously demonstrate that it's due to that protein. But this is intriguing. It clearly says that it's not essential for viability. but we wonder if that increase is hinting at a physiological role nonetheless. So the field right now, there's sort of an emerging view. I would say the data is not fully there to support it, but there are a number of hints that more's not necessarily better, meaning more parasites in the mosquito is not necessarily optimal for transmission. From the standpoint that too many parasites will compete with nutritional resources with the mosquito, will elicit a stronger immune response, and this may be deleterious for ensuring transmission to a new human host. Again, this is sort of an emerging view, and the data is still coming here, but it raises the intriguing scenario that perhaps part of the role for this divergent cytochrome may be as a molecular capacitor to help prevent overgrowth, and that may be part of the selective pressure to retain it. This is sort of an emerging view, and it's one we're looking to follow up more. So just trying to pull all of this together. So despite recognition that the electron transport chain is really one of the key pathways for parasites, these studies are literally the first direct tests of heme utilization in blood stage Plasmodium. The conserved cytochromes C and C1 are absolutely critical for electron transport chain function, for oxidative recycling of ubiquinone to support blood stage growth. And knockout of just that conserved cytochrome C alone, or knockdown, I should say, is sufficient for cellular death. And in this regard, really contrasts with that gene duplication I mentioned is present in yeast. Is there something targetable here? We think it would be challenging to figure out how to directly target cytochrome C itself. However, aspects of the holocytochrome C synthase, I don't have a lot of time to go into those details, but we think there may be some features distinct from the human HCCS that possibly could be exploited therapeutically. We're in the very early stages of evaluating that. Dennis is asking about mass spec interaction studies for C2. Dennis, hold on to that question for 20 seconds and I'll get to it. So just wrapping up then for C2, this is sort of a highly divergent cytochrome C homologue that's been retained. So again, Plasmodium has a heavily skewed AT-rich genome that acquires mutations readily. And so without selective pressure to retain it, that gene would have degraded over evolutionary time. So the fact that it's been retained implies strongly that there's a selective pressure to retain it. And our challenge is to try to deduce what that selective pressure and fitness benefit is. It clearly has molecular adaptations that defy a conserved function in the eukaryotic evolution and in the electron transport chain. We hypothesize that it has constitutive peroxidase activity. We're testing that now. It's clearly not essential in blood or mosquito stages, but does appear to confer, or at least the hints are that it confers a minor fitness advantage in blood stage and may help prevent overgrowth in the mosquito stages. We've done IP mass spec studies on both C and C2. All I can say is they don't allow us to distinguish anything functionally between the two of them. Both come down with members of the electron transport chain, as you'd expect for an intermembrane space protein. We were hoping there might be some smoking gun in terms of selective interactors there. We haven't seen that. Yet. So just wrapping up with the perspective here, we've broadly set out to understand both heme acquisition and utilization. And one of the challenges, but also rich features of Plasmodium biology are these distinct host environments and the metabolic tuning that goes on that are different in the different growth environments that are clearly relevant for thinking about what's an appropriate therapeutic target, what's appropriate for thinking about transmission blocking, but also gives insights into divergent eukaryotic biology and these differing solutions to nutrient acquisition, but also molecular and protein evolution that these divergent eukaryotic cells have undergone. And so, as I said, there's a two-pronged motivation here to enrich broadly our understanding of this fascinating biology, and then think hard about how we can leverage those insights for therapeutic purposes. So I'll close by acknowledging members of my lab. I had highlighted the key people that had worked on this. We have a number of other areas that I didn't have time to talk about today, and our funding sources here. And we'll thank you and take any additional questions you may have.
Great, thank you, Paul. Let me see. Dennis, do you want to ask that additional question?
Paul, can you suppress the essentiality of cytochrome C by overexpression of C2?
Yeah, I think the answer is no. There's one critical control there we haven't done yet, but by overexpressing C2, that is not sufficient to rescue C. That's consistent with all of those molecular changes that suggest that the reduction potential, we do have some preliminary redox potential measurements that suggest that it's massively skewed and thus not biochemically optimized to serve as that mobile electron carrier.
Great.
Looks like Roland has a hand up.
Yeah.
Me?
Yep.
Yeah, very nice talk. Reminds me of my old heme studies.
That's right.
So
Back when the name of that enzyme, the name of that protein was a lot simpler and less of a mouthful.
Yes, cytochrome C heme. So I wonder whether you have an idea what the N and C termini, they're longer in the C2, what they might be doing. So one question would be, are these extensions necessary for covalent heme attachment in E. coli?
Right. So I think the answer is likely to be no to that. We have done some truncation studies that suggest that, smaller portions of that are sufficient for hemolation. That's consistent with recent work from Bob Kranz's group showing that just small peptides, as long as they have that HCCS recognition sequence and the CXXCH, that those are efficiently hemolated in vitro. Coming into what role do those play? We wonder whether those are interaction handles for binding to other protein interactors. One of the challenges here, because we've not yet unearthed a strong phenotype, that it becomes more challenging to deduce what that key cellular function is. As I said, we've done those IP mass spec studies that don't suggest obvious differences from the more conserved cytochrome C. We've started turning to proximity biotinylation studies and other systems that can provide some really rich insights that are highly complementary to traditional IP mass spec studies, and we're starting to move those forward with this system.
Thank you. And maybe a more general question, going basically to the beginning of your presentation. You didn't mention catalase and cytochrome P450s as potential heme-dependent enzymes in your parasite.
So they lack those. They lack obvious types of those. And so that's what I meant by they seem to have an unusually stripped down collection of identifiable heme proteins there. Yep, absolutely.
Thanks.
Yeah, thank you.
Other questions? Okay, well, Paul, thank you so much for the great talk overview of really how you've advanced a lot of the research. It's so much fun to hear and see the progress that's happened on that C2 molecule itself and trying to understand and postulate what it might be doing, which is really exciting.
Yeah, it's been fun. As I said, obviously, is in experimental biology, one feels gratifying when you have some huge dominant phenotype on these things. Of course, you've taken a sledgehammer to something, because those are oftentimes, most interpretable or maybe easy to justify. And, obviously there's hints here of some things going on, and we're hoping to be sort of attentive scientists and keep our nose to the path here and figure it out.
Great, just terrific. If anybody else wants to ask a question, they're welcome to pop in. You can unmute yourselves and just say hello to anybody you wanted to say hello to. And thanks everybody for your attendance. And again, thank you, Paul, for the wonderful presentation. Really, really excellent.
Great. Good to see all of you here. Good to see you, Roland.
All right. Nice to see you too, Michael. Okay, take care, everybody.
All right. Bye-bye.
Bye. Thanks, Paul.
Yep, my pleasure.